Hello guys, this is my YouTube channel and I'm going to provide the notes of biotechnology chapter first, biotechnology principle and processing. First of all, it's important to know that what is biology, what is biotechnology. Bio world is for a living, and technology is the technique which we use. Hence, we use a living organism for biotechnology. Let's move further. This is the definition of NCERT. Biology deals with the process of using a living organisms to produce a product process useful for humans. Generally, we use biotechnology to form the product which is useful for human beings. EFB, remember it? European Federation of Biotechnology. EFB gives definition of biotechnology as following. It is important to know that because sometimes the question arises from this portions also. The integration is sum up. In physics you know about the integration. You sum up the all the differentiations units like integration. You sum up all the things. Integration of natural science and organism cells, parts of thereof, and molecule analogous for product and services. Now biotechnology is useful in various fields, that is in vitro fertilization. Biotechnology is useful test tube babies form now, and development of vaccine etc. Development of vaccine and etc. things we use a biotechnology also. Let's move further. Principle of biotechnology. Generally, principle of biotechnology, there are two main principles, because modern biotechnologists depend upon these two principles. First principle is genetic engineering and second is the bioprocess engineering. Genetic world, you all know about the genetic world. genetic Genetic engineering, the technique to alter the chemistry of DNA, alter the chemistry of DNA or RNA to the RNA to introduce this into the host cells, and thus the change of phenotype of the host cell. Genetic engineering, simply, you take you generally alter the DNA and RNA, and this alter DNA you introduce this DNA into the host cell, and thus the change of phenotype of host cells occur. Bioprocess engineering. What is the bioprocessing engineering? It is the maintenance of sterile. Sterile is a microbial and contaminant free. Ambient, this world is meaning of atmosphere and like place is chemical in chemical engineering process to enable the growth of desirable microbes eukaryote cells in a large quantity for a manufacturing of biochemical products like antibiotics, mm, anti antibiotics, vaccines, and enzyme, etc. It is the principle of antibiotics. And further moving towards, there is asexual reproductions preserve the genetic information, while as a sexual reproductions permits variations. Generally, we all know from chapter first of class 12th, you know that asexual reproductions generally preserves the genetic material. We all know how they preserve genetic material, because there is the single parent is involved. Therefore they form the clone. What is the clone? Clone is the genetically and morphologically similar as a clone. And sexual reproductions permit variations, because two parents are involved during fertilization. So this sexual reproductions permits variations. This is sexual reproduction. Take how the piece of DNA replicate in the host cell. This is all about I discuss in a later upcoming video. Thank you, and please subscribe my channel.